use of an organism or life system to generate a product or process. A common example would be creating antibiotics or drugs such as penicillin to biotechnology is using lab techniques to create products that we see every day. So in the case of antibiotics, that would be the manipulation of our body systems to create prescriptions and medications. Next slide. So a lot of modern biotechnology depends on a critical component of our body, DNA. DNA technology is really, really important for modern biotechnology, and some common examples include PCR, gel electrophoresis, DNA cloning, and DNA sequencing, all four of which we will go over today. Next slide.
Okay, so now I'm going to talk to you guys about polymerase chain reaction. So polymerase chain reaction, also known as PCR, is a method widely used to rapidly make millions to billions of copies of a specific DNA fragment, allowing scientists to take a very small sample of DNA and then amplify it to a large enough amount to study in detail. So PCR is fundamental to much of genetic testing, including analysis of ancient samples of DNA and identification of infectious agents. So using PCR, copies of very small amounts of DNA sequences can be exponentially amplified in a series of temperature changes. So PCR is now a common and often indispensable technique used in medical laboratory and clinical laboratory research for a wide variety of applications, including biomedical research and criminal forensics. So it can also be used in DNA diagnostics. For instance, if you want to check for a gene that could predispose you to do a certain condition, you could conduct a PCR on a small amount of that gene and then amplify it to see if you have enough of it to have that condition. So how is PCR conducted? So in PCR, there are 5 main ingredients required to set it up. So these ingredients include a DNA template to be copied, primers, which are short stretches of DNA that initiate the PCR reaction. So these primers are designed to bind to either side of the section of DNA that you want to copy. They're essentially complementary to the ends of the DNA sequence that you want to copy. You also want to add DNA nucleotide bases, which consists of adenine, thymine, guanine, and cytosine, which are essentially the building blocks of DNA. You also want to add Taq polymerase, which is an enzyme that is used to add DNA nucleotides. And finally, you want a buffer, which consists of water and some minerals and salts to ensure the right conditions for the reaction. And so you would put all these ingredients into a PCR tube, and this entire process is automated and can be completed in just a few hours in a machine known as a thermocycler. So as you can see in the photo, this thermocycler is programmed to alter the temperature of the reaction every few minutes, allowing the DNA to denature and then synthesize. So what are the steps of PCR? So there's 3 main steps of PCR: denaturation, annealing, and extension. So the first step is denaturation. So in order to do the goal of PCR, which is to amplify a specific segment of DNA, the sample is first heated to around 94 to 95 degrees Fahrenheit. So the DNA fragments denature or separate into 2 strands of single-stranded DNA. Because remember that DNA is double-stranded. So the initial goal is to heat up the DNA so that it unwinds into 2 single-stranded DNAs. The next step is called annealing. So next, the temperature is lowered to around 50 to 56 degrees Fahrenheit to enable the DNA primers, which are in that PCR tube, to attach to the template DNA through hydrogen bonding. So primers are single strands of DNA that are about 20 to 30 base pairs in length, and they are designed to be complementary to short sequences of DNA at the ends of the fragment that you want to copy. So typically, if you were conducting a PCR, you would order lots of primer from like a biotech company that is specific to the sequence that you want to copy. The final step is called extension. So during this final stage, the temperature is increased to around 72 degrees Fahrenheit to enable an enzyme called Taq polymerase to add new DNA bases. So Taq polymerase synthesizes two new strands of DNA using the original strand as a template. So this process results in the duplication of the original DNA, with each of the new molecules containing one old and one new strand of DNA. And so this model is basically called a semi-conservative model, where in DNA replication, you have one old DNA molecule, and that serves as the template for the new strand. So then each of these strands can be used to create two new copies and so on, sort of like in a cycle. So this cycle of denaturing and synthesizing new DNA can be repeated to around 30 to 40 times, leading to more than 1 billion exact copies of the original DNA segment. So as you can see in this photo, these are the steps of PCR. You start off with a double-stranded DNA, and then in the denaturing stage, you would heat up the DNA so it separates into two strands. Then in annealing, the primers attach to the single-stranded DNA after lowering the temperature. And then in extending, Taq polymerase attaches to that primer and starts to add nucleotides, extending the molecule to become double-stranded. So some commonly asked questions about PCR that kind of confuse people include, so why do we need to heat up the DNA initially? Well, the key reason for doing this is to separate the strands. Because unlike in your body, when DNA replicates, there's an enzyme in human cells called helicase, which essentially unwinds the DNA. But when you're doing PCR, there is no enzyme to unwind the DNA and separate it. So instead of using an enzyme, heating up the DNA pretty much works well enough to separate the strands. So another question is, so why do we add primers? Well, primers serve as the starting point of DNA synthesis because the enzyme that adds nucleotides, which is polymerase, can only add them to an existing strand of nucleotides. So without that primer, which is a short sequence of 20 to 30 nucleotides, Taq polymerase would not be able to add on the nucleotides and make the single strand into a double strand. So why can we only use Taq polymerase for PCR? Why can't we use a different type of polymerase? Well, Taq polymerase is extracted from a really heat-tolerant microorganism called Thermus aquaticus. This bacteria normally lives in a hot spring and can tolerate temperatures above 80 degrees Fahrenheit. So the bacteria's DNA polymerase is very stable at high temperatures, which means that it can withstand the temperature needed to break the strands of DNA during the denaturing stage of PCR. So DNA polymerase from other organisms would not be able to withstand these high temperatures. Another question is, so what does the chain component of polymerase chain reaction refer to? Because we know that polymerase is the thing that adds the nucleotide. So what does the chain component actually refer to? Well, it also refers to Taq polymerase adding nucleotides because it sort of does this in like a chain fashion. It adds on the nucleotides via hydrogen bonding, and it sort of creates like a chain of nucleotides. Another component of this chain is because we have all the ingredients still in the tube after the first cycle of PCR, you can start the process again and heat up the DNA and go through the steps of PCR, and the two double strands can turn into 4 single strands. And then you can do the process again, and those 4 single strands turn into 4 double-stranded DNAs. So this is sort of a cycle, like a chain, like it keeps on repeating. And finally, how does Taq polymerase know when to stop adding nucleotides? Well, on the first cycle, it typically does not know where to stop. But when it goes in the other direction on the second cycle, it reaches the primer and has nothing else to copy. So when it has nothing else to copy, it just stops. And this essentially results in a billion copies produced with nice clean cuts and similar sizes. So here are a couple of photos of PCR. The first one here shows the different steps of PCR. So here you start off with the target sequence, and then in denaturation, you separate the DNA into two single-stranded DNAs. And then in annealing, the primer is attached to the 3' end of the single-stranded DNA. And then in extension, DNA polymerase adds nucleotides to the 3' end on each primer, creating a double-stranded DNA. And as you can see on the photo to the right, it shows the different cycles. So one cycle can result in 2 double-stranded DNAs, and then the second cycle can result in 4, and so on. So it sort of doubles every time. So here's a short video explaining PCR.
Thanks for stopping by. This is 2-Minute Classroom, and today we're talking about polymerase chain reaction, or PCR as it's more commonly known. First, let's talk about what PCR is and how it works, and then I'll give some examples of what PCR is used for. PCR is a technique used to amplify or copy small specific segments of DNA. For PCR to be successful, you need the DNA sample you want copied, DNA primers to identify the specific sequence of DNA you want to amplify, lots of raw nucleotides to form the DNA copies, and a heat-resistant DNA polymerase to make the new copies. This all goes into a special stable solution. Once all the ingredients are in, you run the solution through many temperature cycles until you reach the amount of DNA copies needed. There are 3 steps to this cycle. Step 1 is denaturation. The solution is heated, allowing complementary DNA strands to separate so that all the DNA in solution is single-stranded. Step 2 is annealing. The solution is cooled so that the DNA primers can bind to the specific region of DNA you are interested in. Step 3 is extension. The temperature is raised so that the DNA polymerase can bind and perform the necessary operation of synthesizing new DNA strands. This cycle is repeated about 30 times, takes about 3 hours, and creates over 1 billion copies. So what is PCR actually used for? PCR has many research-based and practical applications. It's used for DNA mapping, like in the Human Genome Project. It's used to check sections of DNA for genetic testing to determine potential disorders. Companies like 23andMe and Ancestry.com use PCR to run personal DNA tests. And my favorite application for PCR is in forensics. Whenever a crime is committed, the perpetrator almost always leaves their DNA at the crime scene. This could be hair, semen, skin, or even blood, for example. Investigators search for this evidence and, thanks to PCR, can amplify the DNA and compare it to the DNA of suspects. This genetic information is then put into the FBI's DNA database, CODIS. But it gets even better. Investigators have been able to take DNA evidence from crimes committed long before CODIS was put together and run it through the current system. This has solved many cold cases and also exonerated prisoners who had been wrongfully accused, even some that were on death row. Needless to say, PCR is one of the most important scientific advances ever. And now you know why. If you haven't, my best to answer those in the comments below. And don't forget to check out my other great
So here's another video of PCR actually being conducted in a lab.
Okay, today we're going to go through a technique known as PCR, or polymerase chain reaction. This is a technique where DNA is amplified using known primers against a specific piece of DNA. The DNA will be amplified by the polymerase chain reaction, and the products of the reaction can be seen using an iGEM gel electrophoresis. The gel is viewed under UV light, and you can run markers of known size of base pairs, and this can determine the size of the DNA which has been amplified. Okay, there are several reagents which are required for producing the master mix for the reaction. The first reagent is buffer, which provides an optimum solution conditions for the reagents to work. This is added to your reaction tube. The next reagent is magnesium chloride. Between reagents, always make sure you change the tip so you don't contaminate your reagents. Magnesium chloride will be added. Pipettes are used to ensure that you get the correct volume of each reagent. As you add the reagent to your master mix, you tick off your working list that you've added your reagent and that you don't add double the volume that's required. The next reagent will be magnesium chloride. And magnesium chloride helps the enzyme to anneal to the DNA strand. The enzyme used is Taq polymerase, which is added at the end. And the magnesium chloride has been added. Each volume of reagent is known on your worksheet, and the pipette is adjusted to get the correct volume to add to your working master reagent. Okay, the next reagent is the forward primer. The primer is a piece of DNA which corresponds to the piece of DNA that you're actually looking for. And the next reagent is the reverse primer. This is added to your master mix. As you can see, all reagents are kept on ice. The next reagent to add are the dinucleotide triphosphates, and these are the bases which will be used by the enzyme to synthesize the new DNA in the amplification process. After the dinucleotide triphosphates have been added, a volume of distilled water is added to the master mix. And the final reagent to be added to the master mix is the Taq polymerase. Taq meaning Thermus aquaticus, which is the bacterium from where the enzyme has been obtained. And that's added to the master mix, and that's ready to use, that's ready to be added to the sample. Okay, so now we have our 4 tubes ready with the master mix, and all that remains is add our DNA sample to each tube. The tubes have been pre-labeled beforehand, and the scientist just checked the tube to make sure she was adding the correct sample to the correct tube, which is vital when you're doing any assay. And the sample is mixed well with the master mix reagent. Sample 3. And a fresh tip is used each time for each new sample. So now the sample has been added to the reagent, and the next step will be the thermal cycling of the DNA to allow the polymerase chain reaction to occur. Once the samples are prepared, they are then placed in the thermal cycler. The lid is closed and the cycle is started. And the cycler is pre-programmed to reach certain temperature for a certain period of time. The initial temperature is 95 degrees for 5 minutes, and this allows for complete denaturation of the DNA. And then the double-stranded DNA separates and the primer attaches once it's cooled to 45 to 60 degrees. This is where the annealing of the primer and the single-stranded DNA occurs. The cycler heats again to 72 degrees C, where the DNA polymerase amplifies the DNA. And then it is heated again to 95 degrees to separate, denature the double-stranded DNA. This This takes place over 30 cycles. And the final extension is at 72 degrees centigrade for 5 minutes. Once the cycling is complete, the samples are removed from the thermocycler and placed on ice. And they are now ready to be run on an agarose gel, or they can be placed in the freezer to be stored until they're ready to be run on an agarose gel.
Okay, so before moving on, I want to kind of like review PCR a little bit. So could you guys type in the chat, what are the 5 main ingredients of PCR? Correct. Taq polymerase. Water, yeah. What else? Primers. Buffer. Yeah, exactly. So what else did you guys notice about like the video? Did you notice any lab techniques that they used? Any like procedures that they did before moving on each step? You guys want to just type it in the chat? Exactly, switching tips, that's very important. So whenever you like go into lab and do like a certain procedure like PCR, whenever you change materials, like let's say you want to put in nucleotides and then Taq polymerase, you want to make sure to change the tip because otherwise that could contaminate the next thing you put in. So every time you want to put in something else, make sure to change the tip. It's very important. They also had a lot of safety gear. They had goggles, they had gloves on, a lab coat. Icing the sample. Awesome job, guys. Okay, I think we can move on.
Mason, did you want to share an activity?
Yeah, I would. Thank you very much. I'll be sending the link in the chat right now. Okay, so basically, because you guys understand like DNA and DNA matching, and we're talking about forensics and stuff like that, I thought that this would be a good activity to just kind of insert here. And basically what that entails is, once you guys are able to hop on, let me know if you guys have trouble accessing it. Oh, whoops. Give me 1 second. I accidentally put it in the wrong place. Here's the activity. And basically, if you guys want to take a look, this is a forensics activity. So basically what you're doing is you're matching someone with DNA analysis from someone from DNA at the crime scene to different suspects. And I know while this isn't necessarily exactly related to PCR, this is what occurs after you get your PCR results. So basically what happens is they amplify sequences in PCR like you guys were learning. And then they take those sequences and they try to match them with different suspects. And while this is definitely a simplified version and you're not doing the gel electrophoresis or any sort of things like that, this kind of is what occurs. So basically what you're gonna do is for each of these answer choices, you guys are going to take a look at the DNA sequences, and you're going to take a look at the sequence in the glove on the left and the sequence on the glove on the right, and you're going to see if any of those match any of the sequences provided by the suspect. Now, there might not be, the suspect's DNA has to include the entire DNA sequence that is in the hair glove, the hair in the glove. But those sequences can be anywhere in the suspect sequences, just as long as they have both. And that will help you pinpoint who was the criminal in this case. And as you guys definitely know. You know, DNA in this sort of cases are increasing, and after like O.J. Simpson and stuff like that, it certainly has become a more prevalent way to try to find out what happened in specific crimes, and it's become a lot more reliable as they found like other methods of conviction have been less accurate. So basically, I want you guys to take a stab at this. If you need it, if you have any questions or don't understand the directions, then I would like you to just ask in the chat. But other than that, I think that's pretty much it. I'm going to give you guys around 15 minutes, and then we can check in after that. And I can walk you guys through them every single question every like 5 minutes or so. So the order of the bases matter. Yes. Okay. So let me share my screen real quick. Yeah. Let's take a look at this. All right. So can everybody see my screen? Okay. So basically, what you're gonna look at here is you're gonna say, okay, we have A, T, C, and G, right? And then you're gonna look at suspect 1, and you're gonna be like, is there any sequences of A, T, C, and G? And I do see one. I see one right here. It's A, T, C, and G. So as you can see here, this suspect has that sequence within their own sequence, which means that this DNA could have originated from suspect 1. Now, to verify this, we must check for another sequence. And normally you would do this across many sequences, but for the sake of the activity, you're going to just do it off of these two. And in this case, we're going to look at ATCG, ATCG. So it just doubled that sequence. Now, if you look at suspect 1 again, you can see ATCG, ATCG. They have two different sequences that both could produce these sequences. So make sure that it's not the same sequence, but it's two different sequences that are producing this. Because otherwise you could get the wrong person. So basically, suspect 1 is definitely in the running because they have these sequences within them. Let's check suspect 2. So as you can see here, this suspect 2 does have. Wait, let me give you 1 second. Okay, so yeah, exactly. Thank you. That's a really good point. Crystal brought up that they have to be following each other. So it has to be in this exact order. They could be anywhere within this line, within the sequence, but they have to be all together. Yeah. So for example, if you're going to look at this, I found an ATCG here and an ATCG here. And suspect 2 is also very much a suspect in this case because they also have this piece of DNA or this sequence here. And they also have another sequence, ATCG here and ATCG here. Remember, it doesn't matter where they're placed. It just matters if they do have the sequence. So I'm going to give you guys, I'll let you guys try for suspect 3 and let me know what you guys get. Okay, so for number 1, suspect 3 didn't do it. So let's double check your theory. So as you can see here, you're very right in that there isn't any ATCGs, and you're also very right that there isn't any ATCGs. So therefore, I believe you are correct. And suspect 3 did not do it. So as you can see here, who committed the crime? We're currently between suspect 1 and suspect 2. And if there's sufficient evidence to prove it, then it would go one way or the other. It's also important to remember that some people, if you don't have enough like DNA, that could also like make it a wide array of suspects. So you want to get as much as possible, and usually you can. But even if they might have been working together even, but that's less likely. So let's move on to question 2. If you're looking at the sequences here, I think you guys should get one definitive answer in this case. Yeah. Once we have our answer, can we check the answer sheet? I do not encourage you to click on the answer sheet, but you can if you want. Yeah, so basically, if you guys want to check the sequences real quick, and I'll check back in like 3 minutes, or actually I'll just wait until somebody answers the question for question 2. Suspect 2. Okay. Yeah, so let's take a look. So suspect 2 does seem very suspicious. So we have CG, 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 CG. 1, 2, 1, 2, 1, 2, 1, 2. Yep. So that they definitely have that one. And CG, CG, so it's just the exact same sequence. And you can see that they also have another one on the opposite side. So great job. And as you can see, for both the other ones, they have A's and T's mixed in there too often. And therefore you can know that they definitely do not match the sequence. So yeah, suspect 2, great job. Okay, let's try number 3. So as you can see here, we have TTTG and TTTC, TTTC. So if you guys want to take a go at that, yeah. And remember, the reason why we do these forensics analysis is because then we can place this person as being at the scene at one point, rather than, you know, just saying that they are. And while we do, of course, like sometimes you can't get DNA, this is the most absolute form of evidence we can get. Especially as the technology has developed, this has been a far more surefire way of placing someone at a certain scene at a certain place in time, which kind of helps. Okay, so you guys said suspect 2 for number 3. Okay, so you guys both agree on it. Let's see. So I think you guys are correct. As you can see here, we have a TTTC and we have a TTTC, TTTC. So therefore, suspect 2 is definitely the person here. Okay, one more, and that's gonna be this sequence with GTA and GTA, GTA, GTA. This person definitely likes GTA a lot. Take that as you will. But let's try to match that sequence real quick. All right. So suspect 2. Okay, let's take a look. Is this guy really sus or no? Okay. So basically we're looking at GTA. So as you can see here, they do have GTA in suspect 1, but it's not long enough. Or yeah, and or no, they have GTA, but they don't have the single GTA sequence. And then for suspect 2, they do have the long enough GTA sequence on the left, and they do have that one GTA sequence on the right. And suspect 3 does not have a long enough GTA sequence on the left and does not have a GTA sequence on the right. So therefore, they cannot be the suspect either. So great job, everyone. That's pretty much the activity. I hope you guys enjoyed that. It was just pretty simple, but yeah. Okay, I'm going to hand it back to Natalie now, I believe, or Joanne, and then they can continue the presentation.
Okay, can everyone hear me? Yes? Okay. So now that you guys have talked, we've learned a little bit about PCR and some of its applications. Actually, I think it's really cool that you guys got to learn a little bit more about PCR. It's super, super important in just scientific research and in bioinformatics and synthetic biology. And actually, a fun fact about PCR is a variation of PCR called RT-qPCR, I think qualitative, or it's either qualitative or quantitative PCR is being used in COVID-19 testing. So that's really interesting. But aside from that, now we're going to dive into gel electrophoresis. This is a bit of a simpler topic. I'm sure most of you guys are familiar with gel electrophoresis or have heard about it at some point in your biology classes or just in reading or anything like that. But gel electrophoresis is a biotechnique used to separate DNA fragments according to their size. So I'm going to quickly run down the first couple of steps of running a successful gel electrophoresis, and then I'm going to talk more about each individual step and more in depth. And then I'm going to also talk to you guys about DNA, so you can have a deeper understanding of what exactly we're looking at here. So firstly, DNA samples are loaded into wells or indentations at one end of a gel, and an electric current is applied to pull them through the gel. So if you're wondering exactly why we apply an electric current and how DNA fragments are actually able to move through the gel, it's because DNA fragments are negatively charged. And I'm going to show you guys a deeper image that shows the individual components of DNA so you can see where this negative charge comes from. But DNA fragments are negatively charged, so they move towards the positive electrode. So in chemistry, you may have learned that we like to say opposites attract, or a positive charge and a negative charge will attract each other, while two positive charges and two negative charges will repel each other. So since DNA is negatively charged, it'll move towards the positive end of the gel electrophoresis gel. In addition, because all DNA fragments have the same amount of charge per mass, small fragments move through the gel faster than large ones. And also, when a gel is stained with the DNA binding dye, the DNA fragments can be seen as bands, each representing a group of same-sized DNA fragments. Okay, so now we're going to take a closer look at DNA before talking more about the steps of gel electrophoresis. So firstly, DNA takes on the shape of a double helix. A little bit of history here. The double helix was discovered by the scientists Watson and Crick. You guys may have heard their names before. They're pretty catchy names, all the way back in 1953. And I would highly suggest taking a look at the history of how DNA was founded. If you're not familiar with it already, there's a little bit of tea, a little bit of drama with it. So I would encourage you guys to check that out. But eventually, Watson and Crick would earn a Nobel Prize for the discovery of the double helix shape of DNA in physiology or medicine in 1962. So in the center of your screen, you can see a blown up plastic model of what DNA looks like. We call it a double helix because you can see these red things, sort of like there's two strands of red on the outside. So that's where double comes from. And we call it a helix because it takes on the shape of a descending spiral staircase. So DNA is made up of 4 nitrogenous bases as well as a sugar phosphate backbone. So the nitrogenous bases take the name of adenine, thymine, cytosine, and guanine. And usually scientists will shorten this and we'll just call them A, T, C, and G, because otherwise it's sort of a mouthful. But in the image on your screen, you can see that these bases, A, T, C, and G, are located in the center of DNA, and they make up the rings if you're looking at it like a ladder or like the steps of a staircase. So purines, A and G, are what we classify as purines, which means that these nitrogenous bases are double ringed. And C and T are what we classify as pyrimidines, which means these nitrogenous bases are single ringed. And the way I always remember this is that purines is the shorter word, but in fact, purines signifies that there are 2 rings. So it's like opposites. While pyrimidines is a longer word, but it signifies 1 ring. So that's just how I remember it. And finally, it's important to note that A and T pair together like puzzle pieces, and C and G pair together like puzzle pieces. So again, on your screen, you can see that purple and green are adenine and thymine, and they're always going to be next to each other, always connected to each other. And the same goes for red and blue, cytosine and guanine, which will always be connected together. And if you're wondering what connects them, it's a hydrogen bond. And this is a really unique quality or an important quality of DNA because this allows us to unwind DNA because hydrogen bonds are strong enough to break, but we can also put it back together because hydrogen bonds are strong enough to keep two things together. I also want to talk about this like tan yellowish thing that's going around the outside, these two tan yellow like lines. This is what we call a phosphate backbone or a sugar phosphate backbone. So if you guys take a look over here to this image on the right side of your screen, you can see that DNA is made up of nucleic acids, and nucleic acids are what we classify as macromolecules. And nucleic acids are composed of smaller little nucleotides. So nucleotides make up nucleic acids, and nucleotides are basically adenine, thymine, cytosine, guanine, plus this phosphate backbone. So here you can see it in another picture. You can see the bases we talked about, A, T, C, and G. It's that little guy right there. And we also see this 5-carbon sugar called the deoxyribose. And this deoxyribose actually gives DNA its name. Deoxyribose nucleic acid is what DNA actually stands for. So we have this 5-carbon sugar, and we also have this phosphate group. So if you guys remember earlier, I said that DNA is negatively charged, and this phosphate group carries a negative charge, which in turn causes the entire DNA molecule to carry a negative charge. So it comes from this phosphate group right there. Okay, so now that I've talked to you guys a little bit about DNA, I'm going to pass it on to Joanne, and she's going to talk to you guys about the steps of gel electrophoresis.
All right, so if we look closer at what gel electrophoresis actually looks like, well, there's a gel, but more specifically, the gel is made out of a polysaccharide called agarose, which when heated in a buffer, forms the squishy substance. Yeah, agarose like agar. It forms the squishy substance that you can see in the image on the side. And the reason why we use agarose is because it forms pores large enough for nucleic acids and proteins to travel through the gel. In the image, we can see that there are pocket-like indentations called wells, which are where you will pipette the DNA samples into. And then at either end of the gel box, there is a power source, which creates the electric current so that DNA can travel from the negative end to the positive end. So Natalie mentioned this a little bit, but DNA and RNA are both negatively charged. So because they're, as we saw in the last slide, there was a negative side and a positive side to the gel box. And since the DNA is transferred into the wells, which are on the negative side of the gel box, once you turn on the power to the gel box, then the DNA will travel from the negative to the positive side. And one well you will reserve for a standard that contains DNA fragments of known lengths. And because logically shorter pieces of DNA will travel faster, that's how you can distinguish the fragments of DNA. In this picture, we can observe the process of gel electrophoresis. First, you load the wells with DNA samples, then you turn the power on, and the DNA fragments will move from one end to the other, with longer fragments staying closer to the negative side and shorter fragments moving farther away from the wells. So there has to be a way for us to visualize the DNA fragments. So we stain the DNA with a dye that will allow it to fluoresce under UV light. And a commonly used DNA staining dye is ethidium bromide, I believe. And And it allows us to see, like you can see on the picture, different lines of DNA, and a well-defined line of DNA is called a band. On the left side, I believe that is the DNA ladder being shown, which I mentioned was the standard that the rest of the wells are being compared to.
So now that we talked to you guys about what PCR can be used for, these are a couple of things that gel electrophoresis can be used for. You can see some similarities between them. But firstly, we have, among other examples that I won't talk about, but we'll have identifying individuals. There's also parental testing, DNA profiling, and fingerprinting. So when we talk about DNA fingerprinting, we're just referring to the fact that all individuals have their own unique individual set of DNA. And you might be asking, Natalie, what about twins? But even twins that are biological clones of each other have DNA differences due to environmental factors. This phenomenon is covered by the field known as epigenetics. So epigenetics just talks about how external factors can cause DNA or gene expression differences. So DNA fingerprinting would just be like we can identify individuals based on their own unique set of DNA. So that can go hand in hand with criminal investigations. We can compare DNA at one crime scene and take samples of suspect DNA and then compare them and possibly find the culprit. And we can also study evolutionary relationships by analyzing genetic similarities between organisms. So again, in the activity you guys did earlier, we could see how scientists can isolate individual bases, A, T, C, and G, and we can see similarities between different organisms. So we can compare the base sequences between different organisms, and those base sequences translate to gene expression, so we can see gene similarities between organisms, which helps us in evolutionary biology. So finally, the last couple of things we're going to discuss today are DNA cloning and DNA sequencing. So yesterday, you guys received a presentation on plasmids and DNA that kind of gave you guys a quick overview of what DNA cloning is and what we use it for in science. You guys should have seen this picture on in your presentation yesterday in one of the earlier slides. But just a review, in DNA cloning, researchers make many copies of a DNA fragment of interest, such as a gene. In many cases, DNA cloning involves inserting a target gene into a circular DNA molecule called a plasmid, and this here is a plasmid. It's exactly what it sounds like. It's just circular like this. So this is a pretty accurate picture. It's not much more complex than this. But the insertion is done using enzymes that cut and paste DNA. If anyone can post the name of the exact enzyme that cuts and pastes DNA in the chat, that'd be brownie points for you, if you can remember it. I think it was discussed yesterday. If it wasn't, then I'll tell you guys what it is when I look in the chat. But there is an enzyme that cuts and pastes DNA, and it'll produce a molecule of recombinant DNA. So this right here would be a plasmid, and then after we cut and paste it using the specific enzyme, we'll get this ring structure here, which is a recombinant plasmid, which just means it has foreign DNA inserted within it. And I think you guys yesterday learned a little bit about how we use plasmids and bacteria. So we can use plasmids, we can give bacteria like a heat shock, and this can like kind of temporarily denature the semi-permeable membrane of a bacteria, and thus the plasmid can sort of diffuse inside the bacteria, and thus when the bacteria grows and proliferates, we'll have DNA cloning occurring. And then we can also have a process called transformation, where we can actually change the property of bacteria by inserting foreign DNA via a plasmid. The chat is popping off right now. Let me take a peek before I talk about DNA sequencing. Oops. Yep, endonuclease. Yeah, amazing. Yes, and someone said ligase. Okay, so endonuclease is what we use to cut and paste, and then ligase is sort of, it's like a glue. Ligase is an enzyme that acts like a glue. So yes, that's completely correct. I was going to say something. Oh yeah, and also, I don't know if they went over this yesterday, you guys might already know this. Whenever you see like a name of something that says A's at the end of it, ASE, it's going to be an enzyme. Sounds like a little hit, like endonuclease, ligase, polymerase, those are all types of enzymes. And enzymes are just proteins that help in catalyzing reactions. Okay. So this is a brief explanation of DNA sequencing. So DNA sequencing is a laboratory technique used to determine the exact sequence of bases, A, C, G, and T's in a DNA molecule. The DNA-based sequence carries information a cell needs to assemble protein and RNA molecules. So if you guys know, so I talked to you guys earlier about DNA and what it's made up of, and I also mentioned for a brief second that we can actually open up DNA, and there's a process that happens within a nucleus of a cell that's called transcription, and we can open up DNA, and we can basically different enzymes will go across the DNA. There's a whole ordeal with leading and lagging strands. That's really interesting if you guys want to look up a video on transcription. But basically the goal of transcription is to make mRNA that will go into the cytoplasm of a cell, and then eventually we'll make protein from this mRNA. So DNA directly codes for proteins / genes in organisms. So if we know the exact sequence of bases, A, C, T's, and G's, we can kind of know exactly what bases and what sequence of DNA codes exactly for what proteins and what genes, which is really helpful information. So as I said, DNA sequence information is really important to scientists investigating the function of genes. The technology of DNA sequencing was made faster and less expensive as a part of the Human Genome Project. In one of the videos you watched earlier, they did mention the Human Genome Project, and I'm sure many of you guys have heard about this before, but if you haven't, the Human Genome Project is basically this giant project that researchers and scientists have been doing for a while, and they have this huge database where they've been conducting lots of DNA sequencing, and then they would save all this information on a computer, and so we basically know for research purposes and for scientific and for evolutionary biology purposes, what exact sequences code for what exact protein. So we have all of that harbored in the Human Genome Project, which is really, really, really helpful for studying science as it involves genes and DNA and such. And also, I forgot to mention that DNA sequencing is more like a broader term. DNA sequencing can be carried out through a variety of mechanisms. So I'm going to give you an example of one. This is Sanger sequencing. It kind of combines lots of things we talked about today. Sanger sequencing is also known as the chain termination method. So as you can see on your screen, firstly, we start with PCR. So we're going to amplify lots of DNA. We're going to make lots of copies. So PCR with fluorescent chain terminating ddNTPS. It sounds complicated, but that ddNTPS is essentially a fluorescent dye that we can use to tag each of the bases with a separate color. So you can see on your screen that G is yellow, A is green, C is blue, and T is red. So we have like these, we color code them almost. And then in step 2, we'll see size separation by capillary gel electrophoresis. So we'll run these DNA samples through a gel, and we'll separate each of them into bands, as you can see on your screen. And then we can shine a laser beam through the bands that we have separated through gel electrophoresis. It'll go through a photomultiplier, and it'll be outputted on a chromatogram. And then you can see exactly, and on your far right of the screen, the step 3 laser excitation and detection by a sequencing machine. We can see the exact sequence of bases that we have on our tiny little piece of DNA. So that's just one way we can DNA sequence. So that is all we have for slides today. But of course, I'm going to open it up for any questions, any comments. Any of us can answer anything that you guys might have an inquiry about. I hope you guys enjoy learning briefly about really important biotechniques that you guys will definitely come across if you decide to take like AP biology, or if you have taken biology, or just in regular biology and in chemistry. And if you decide to do your own research projects, all of these techniques are very helpful to have some introduction to.
Yep. And also, we do have a little bit of extra time, so would you guys like to do another activity? I could send another activity here. Oh, wait. Okay, so I don't have a Kahoot per se, but I can look for one. If you guys, do you really want to do Kahoot? Is that like your goal in life, just to do another Kahoot? Okay, let me look for one.
You guys like Kahoots? I like Kahoots too. Wait, Aaron asked me, this is totally off topic. I'm not going to read out loud, Aaron, don't worry, but I'll answer your question. What is it? Do I have to? So, okay, while Mason's finding a Kahoot, I'm going to answer Aaron's question. You guys could ask me random questions too. I can answer anything. So, Aaron, I'm in theater conservatory, so I'm a senior in theater conservatory. So the answer to your question is no. If you want to participate in schools / musicals, you don't have to be in theater conservatory. But I will say, unless I don't think this, okay, I'm going to say this with confidence. If you audition for a play at CCA and you're not in conservatory, you're probably not going to get the best part, not because you're not talented, but only because they prioritize conservatory members. So that's the advantage of being in conservatory. That's why I'm in conservatory. So when you're in conservatory, you're guaranteed, first of all, we do lots of workshops, and we have like a community together, and we practice when we're not doing musicals, but you're also guaranteed a part in every single musical or play that you audition for. So you're not gonna never not get in if you're in conservatory. And you're also guaranteed like an understudy part or some lines. So that's an edge of being in theater conservatory. Oh, it's hard for you to balance swimming, quiz bowl, and conservatory. Thanks. Yeah, I totally understand that. Yeah, theater conservatory does take a lot of time. I think out of all the conservatories, it takes the most time, but I would obviously recommend it. I loved it. I'm still in it. Yeah, I do a lot of dance outside of school, and I do a lot of science ECs too, but yeah, definitely it's really hard to balance sometimes, but I have faith in you, and you can still audition for anything you want, even if you're not in conservatory. Okay. Did they have a gene sequence? Don't they have a gene sequencer at CCA? No, question mark? Maybe. You heard something about it? Yeah, there's lots of cool stuff at CCA, but oh, for Quest, I don't know. Yeah, Quest is. I'm transferring from You're transferring from Torrey Pines. For science clubs, oh my goodness, iGEM. Yeah, science clubs, I highly recommend iGEM. Oh my gosh, there's so many science clubs. There's so many science clubs at CCA. People love science. Yeah, that's on your list. Okay. You can also make your own science club on anything you like. That's always fun. There are so many science clubs in general. If you go on CCA's website, there's a list of every single club that we have, and we have so many. So I know that there's like the Red Cross Club, and I don't know, there's so many.
Yeah, I looked on the ASB site and like for the club list, and there's like so many clubs to look at. But yeah, I was definitely thinking about iGEM, but I don't know if I did PsyOLE last year, but it's a lot of time commitment. Mm-hmm. And like, since it's going to be my first year at CCA, and I'm like, I don't want to take on that much time commitment because like, because like the schedule's kind of weird and stuff. Yeah. So I want to get used to that, like how like we have to finish 1 year classes in a semester first before I like do like Science Olympiad and iGEM, you know, but.
Right, exactly what you mean. So I'm going to tell you that CCA does, like we do have like a weird schedule because it's like an academy. So we have the 4x4, which is different than Torrey Pines, but I can guarantee you that you will have time to do ECs. If anything, I've had so much time to do extracurriculars, but I think that's a really good idea to just like gauge your schedule, get used to the courses, and then you can sign up for any clubs. People are so welcoming here, so you can sign up in the middle. I don't think anyone would care at all. Or yeah, I definitely think you'll have time. I wouldn't be worried.